Imagine having everything you need for your microscopy workflows in one place, with a sample protecting incubator, all with a single push of a button. Well, you don't have to imagine anymore. And on that note, I'm thrilled to be welcoming you to our webinar, Discover a New Era in Microscopy. And we will be doing just that by introducing you to Mica, the world's first imaging microhub. Our guest today is Svenja Kunnert, Advanced Workflow Specialist at Leica Microsystems. She will be taking you through an experience like no other by speaking about how microscopy has changed with the arrival of Mica. So let's get right into it. Thank you so much for joining us today.
Now I would like to invite you to step with me into the Microhub era, because Mica is a microhub. The questions that you scientists answer are more and more complex and lead to even more data and more questions, right? So it appears that to achieve the breakthroughs that are behind these questions, the data points, need to be put into context. So it is not only important to discover a protein, for example, but also to put it into its spatial context. So where it operates and what's around it. This is the megatrend of the so-called spatialomics, and what is better than insight generation to support that? So come with me and step with me into our microhub and meet Mica. Conventional microscope is usually built for one specific task and requires expertise to use optimally. Mica creates a complete new reality in that. It gives access for all with no constraints and offers a radically simplified workflow. So Mica is a single, easy-to-use digital imaging system that will confidently lead you from setup to relevant insights on a radically simpler way than any other traditional microscope you would use today. Let's go through each of these elements and let's shape the new reality and see how it will positively impact your daily work. So Mica gives access for all. Access for all, that means that everyone, so you as a professor or as a master student, can leverage microscopy to make more discoveries. So we are talking here about eliminating over 85% of the most tedious setup steps that require spatial expertise for setting up an experiment with a microscope. So let's take this example of a 3D tissue image. And you see here the image acquired with a conventional microscope on the left and an image acquired with Mica on the right. When setting up your microscope for imaging, you go through an iterative process of multiple steps so to get the first image. So setting up fluorescence channels, exposure time, intensity, and so on. And this is not even thinking about making the final image acquisition. So these numbers of steps increase exponentially as you add more labels. Mica, compared to a conventional microscope, reduces these numbers of steps over 85% until you reach the first image. So this is a third less time than what you would usually need to get your first image. And also Mica's ease of use reduces by half the time needed to be trained on working independently. So in summary, you tell Mica about your sample and the conditions you want to image, and Mica will set it right for you, leading to comparable or not even better image quality maybe, as you can observe here. And this is all powered by intelligent automation and intelligent illumination embedded in Mica. So let's have a look at the real thing. Let's go live to Sasha at the EMBL in Heidelberg. Sasha, it's up to you.
Thank you, Svenja, and everyone, welcome to EMBL Heidelberg. My name is Alexander Pospiech. People also call me Sasha. But we are not here because of me, but because of Mica, which is right behind me. As we already mentioned, Mica is a microhub, and I will not talk about the features too much. I want to show you how easy in real life it actually is to use Mica. So let's go to the instrument. I will start with mounting the sample. I'm just putting on gloves. I have a living cells there. As I mentioned, Microhub offers several modalities, but also environmental control. Let's look at Mica and how it looks like inside. So this whole box encompasses the microscope and provides the environmental control. With one button, we can unlock it and we can see what's inside. Motorized stage with sample holder and also some extra space where, as you can see, I have another plate, another chamber slide. There is another possibility to get inside of Mica, and that's the smaller window right over here. And if I want to quickly access my sample, I can do it like this without ventilating the whole space. So I can take out the whole sample holder. There are standard, there are various types for Petri dishes, slides, multi-well plates. So you choose what you need. And here I have my 8 chambers. I will put it back in. Okay, this is also, if I have a living cell experiment, I can very easily just pipette, add some extra medium, some treatment, and quickly start the experiment this way. As you can see, the whole inside is black, so I don't have to worry about having the light in the room for fluorescent imaging. And let me show you how do I actually start setting up my experiment. So what do I need? Do I need to have a PhD in physics? Do I need to have 2 years training on software analysis or application specialist on the phone or either sitting next to me so that I can start using Mica and getting nice images? Not really. I need to know information about my sample. What is my carrier? So I'm here. In my case, it's a multi-chamber with 8 chambers. There are options for multi-well plates, Petri dishes, slides, and now characteristics of the sample. So I'm using this detector. I have my living cells there. They are living, and I also have the environmental control on. As you can see now, it says set points not yet reached because I had it open, so the CO2 will be getting to the desired level. And this is the important part. These are my dyes. So I need to know the dyes exactly which are in the sample. Either I prepared it, or for me, my colleague prepared it from Mannheim, Sanjeev Kumar, and these are four dyes staining DNA, mitochondria, tubulin, and lysosomes. And you can see that there is a great amount of the dyes. And if I'm missing some, but I know the spectral properties, I can also add a new one if needed. Mounting medium, of course, is important. And this is pretty much it for me to start imaging. You can see already the template of my carrier. I can select all the chambers if I want, but I only have cells in these two. So I will start with sample overview. What is happening right now is Mica just go in X and Y with a low magnification objective and detects the border of the carrier. This is important for the precise alignment. And now it's just checking whether everything is okay. And right after that starts the quick overview. We have a low magnification objective, 1.6x, with a special contrast that will help us visualize even transparent structures like the cells. or other unstained structures and specimens. Focus was found, sample detection was finished. I'm keen to get my fluorescent image, so I will just confirm and go to live. So now we are switching to live acquisition. And my fluorescent channels are already in. I want to see all of them in at once. I have my 10x objective. Now I'm in wide field. So let's see how difficult it is to get a nice fluorescent image. And immediately I have my 4-color image. The sample is in focus. Mica already took care of that in the beginning. What you could notice now was this intelligent IQ illumination. I'm talking and nothing is happening in the sample. Cells are living, and if I was talking for another 15 minutes and I would keep the light on, I would bleach them, or definitely they wouldn't be happy about the situation. So what is happening now? All the lights are off. I'm seeing the image from the last image basically. I will switch to 20x. And you see that the image is live again. Let's magnify, and this is my nice multicolor simultaneous image immediately. What I can show you as well is I went straight to fluorescence. So you see, I did not worry about the exposure time. I did not worry about the intensity of the light, preparing setting filter cubes or anything like that. I just went live right after detecting the carrier, and I got my image. In the background now, you can maybe hear Mica. So it's pumping the water for the humidity, for the environmental control. So this was straight, my fluorescent image, but we also have a very nice bright field method. So first, the standard bright field, of course, which would look like this. They are unstained, so it's not so easy to see something so clearly, but with intermodulation contrast, I can very nicely see the structures around the cells, navigate. Maybe I will find something more interesting. The benefit of the intermodulation contrast is that it also works with the plastic bottom. And this was just a very quick display on how difficult or easy actually it is to get a fluorescent image. If I want to change the settings, what I have here is field of view. Of course, sometimes I'm interested only in something smaller, so I can decrease the field of view. I can also change the exposure time. So there are some presets. I can change it myself to some specific value that may be relevant to the events I want to observe, maybe in the living cells. I set the resolution. It's in simple settings, slow and high, medium for the wide field, and then image quality. So this is important. So maybe it's stable sample, maybe it's fixed, so I can go for very high image quality, I can image longer. It does not really matter, the sample will be okay. Or I'm interested in capturing some living events, so across a span of several hours, so I will go more for the sample protection. And this is basically, this is all I need to know to start nice imaging. And with this, I will hand word back to Svenja, and she will tell you a little bit more about another features of Mica.
Now let's have a look at our philosophy of no constraints. The era of no constraints is an era where everything you need to enable all your discoveries is unified in one easy-to-use system, This is allowing you to generate 4 times more data 100% correlated. So what does it mean, 100% correlation and 4 times more data? Let me show you what I mean here within this experiment. In this experiment, we labeled U2OS cells with mitotracker green, so shown in cyan, and TMRE for active mitochondria in magenta. So we acquired the images live on the conventional microscope on the left and with Mica shown here on the right. The challenge is to image the different channels first, avoiding crosstalk and bleed-through, for sure. And second, make sure that in moving samples, the correlation of the signals in the 2 channels is precise. So at a first glance, the image on the left acquired on the conventional system looks fine. But when you start looking at the fast-moving elements, you see that the sequential acquisition leads to a mismatch between the 2 labels. So you have a spatiotemporal mismatch between the functional and the structural information. So when filters for the different stainings are used, the sequential acquisition leads to a mismatch between the 2 labels. So this is where Mica's breakthrough innovation shows itself. The 2 channels are imaged simultaneously so that you achieve 100% correlation in space and time. So Mica allows you to image up to 4 labels simultaneously, which provides you true correlation in space and time according structural and functional information. This critical feature of Mica is powered by FluoSync, our patented unmixing technology. Another aspect of no constraints is that Mica brings confocal and wide field imaging seamlessly together. You can use the right modality at the right time and can switch from one to the other. Often you would want to use the fast wide field imaging to generate overviews and then go to the highest detail using confocal microscopy for the extra resolution and contrast. When looking at the spheroid, like the one shown here, you can observe its growth using gentle thunder imaging and go seamlessly to the creation of a highly resolved confocal image for the final recording. And with Mica here, there are no more complications during sample transfer from wide field system to the confocal system, or not by having the access to the confocal in your facility when you exactly need it. Or another example, when imaging an intestine slice like this one shown here, the overview and selection of area of the interest are acquired with wide field and thunder in 20x, before moving to 63x and confocal to visualize the fine details and subcellular information with lightning. But Mica is even more than that. It will be your best partner for live cell imaging. Mica is also an incubator. It maintains physiological-like conditions through your whole experiment. So Mica takes care of stable temperature, gas concentration, and of the humidity, reducing evaporation and the subsequent changes of the concentrations in the medium. So the health of your sample is no constraint anymore. Let's see some of these applications and possibilities in action right now at the Amble system. So, Sasha, up to you.
Thank you for explaining the other features, Svenja. Let's check them out now. I'm at 20x, 4 channels. Maybe I want to check what's happening around. Maybe I want to look for more interesting cells to image. So I will create a region of interest, maybe about this size, 70 tiles. That sounds about right. Let's start imaging right away. This is the speed of simultaneous 4-channel imaging. So you see it's super fast. Even for very, very large areas, I can get very quickly overview. And this is already high resolution, high numerical aperture, 20x objective confocal, so it's a very good one. I can zoom in, for example, if I'm interested already, I can start browsing. The amount of details I'm getting is pretty good. On the background, we did quite a lot of data. 70 tiles, 4 channels, that's how much, 280 images, high resolution images. And in the background, these images were merged already for me. So let's see how it looks like. This is the montage. What I want to do now, maybe I can take it a little bit further. So let's Thunder it. I can go to processing, and I will use the instant computational clearing, which is Thunder. Thunder removes the out of focus blur. So white field typically suffers with the amount of light we don't really want to image, and Thunder is a computational method which reduces this blur, so I have a higher contrast, better signal to noise ratio. And I can Thunder the data in post like this, or I can do it during the acquisition already. I also have the benefit of keeping the raw data. If I want to come back to them, I can compare them or maybe process them with different parameters if I want to play with it. And if I want to do just a quick comparison, this is how I would look at it. So on my on this side on the left is white field, and on the right is Thunder data. So this was my 20x overview with Thunder. What can I do next? Maybe I already, if I can see some interesting cell, yeah, maybe I like this one. And let's go straight to confocal. So now I will grab the sample and I will quickly run over the corridor. We have another system there. I will show you confocal. So, of course, I'm not going to do that. Mica is a white focal. So what I will do first is high magnification. I will use high magnification objective. So this is my 63x. And right after I have higher magnification, I will move to confocal. What is happening right now? Water immersion process active. There is nobody hidden over there pipetting the water on the objective. This is Mica basically taking care of that for me. Of course, it's great. I don't have to go there. I don't have to worry about it. But another benefit is I can rely on this water immersion, automatic water immersion, for example, for a larger tile scan or for time lapses. I do overnight time lapse, and Mica will simply keep refilling the water for me. So I did first thing. First step, I wanted to go for higher magnification. So now this is my 63x. There it is, our cells. Let's refocus like this. Okay, but we are still in white field, and I said I want to do confocal. So this is how I will switch to confocal now. And I can also zoom in because I'm interested maybe just in this one cell. This one cell is good enough for me. Okay, I will check the focus. Actually, just for focusing, I can easily use, I could also use the fast mode, which is more sensitive to a specimen. What I will do, I know that the tubulin channel is very weak, and I think it will be better if I acquire it sequentially. So let's go for this confocal grade, image quality, we are in the middle, they are living cells. one touch. Let's check if the light, if the illumination is okay. Okay, this looks good, and I want to have my confocal image. So my 3 channels, now it's acquiring the last channel in the sequence. And let's check it out. Okay. And just like this, I have my confocal image. I can also improve it further. I'll apply lighting, and let's see how the image can look like. So super simple few clicks, and I got my confocal image. Well, maybe it's still not enough for me. Maybe I'm more interested in 3D, for example, here. Let's check Thunder. What Thunder can do for me, what information I can learn. Let's go, I will put my channel back in. Okay, this looks pretty good. What I will do, I will create a quick Z-scan. Let's activate the 3D imaging. And let's start over here. Then this will be my Z-scan. They are relatively thin, so let's just go on with this 2 microns. And live IQ switching again to virtual live. So this is my last image. The lights are turned off now. Cells are not being bleached. Let's start this 3D Z-scan. That's it. My Z-scan is done. All channels were done all channels were done simultaneously. I want to see what's the best I can get out of this image, and that's why I will go for Thunder large volume computational clearing. This one is better for 3D information. Okay. My Thunder is finished. And here we go. White field and Thunder. These are living cells. This is not like perfect fixed sample. They are alive. So, what do you think? How do you like this?
I would like to show you right now how far you can go, even until 5 p.m. with our radically simplified workflow. So the era of radically simplified workflows is an era where we bring you faster from sample to discovery. So Mica will reduce over 60% of process steps through system intelligence. On the left, here you see the various different steps, from sample mounting to analysis, that you need to run when you set up an experiment on a conventional microscope. So we have counted 24 steps for a typical multicolor experiment. When using Mica on the right, this workflow is radically simplified into as little as 8 steps, reducing time and efforts needed from sample to the insight. This simplification is also used through the entire analysis process. Simply by painting on the image, you can tell Mica what is background and which are the features that you want to measure your structures of interest. On the base of AI, so artificial intelligence, Mica will automatically train its machine learning pixel classifier and identify the parameters required for the segmentation. Then you can select which values you want to compare, like intensity, diameter, form factor, and so on. And you can directly create a visual representation, whether it's a scatter plot, pie chart, or graph, whatever you would like to have. For sure, the model generated by your training can be exported and shared, ensuring 100% repeatability and reproducibility. So you can also reuse existing models and enhance them by further training. I think, Sasha, you need to show us this completion of the workflow with Mica.
So let's look into these options. Of course, with the standard imaging, we have all these traditional options like creating Z-scans in the wide field. This is very fast, it's sensitive for the sample. We can do the same with lightning, and we have a beautiful, detailed, confocal super resolution, just like that. And the same thing I actually did with living cells. You saw that Mica is also incubator. This is just a quick Z-scan I created with Thunder on live cells. So it does not matter whether you have fixed samples or living cells, you can really benefit from this easy of use. And now it's time to move from imaging to image analysis. So we do have built-in machine learning image analysis in the Mica LASX software. And with this option, Learn, we can load a data set. Let's start creating a completely new model. So I will call it, we will look into detecting mitochondria, so I'll call it my mitochondria, for example. Now, Mica again is guiding us to what we should do next. Add an image. So I have here a nice confocal lightning image I made on the living cells. And now I will play a little bit with the options we do have here for the software training. So I need to specify what actually are my objects of interest. So I want to look into the mitochondria. So I already have that labeled from the very beginning of the experiments. When I was setting dyes, I was labeling which structures they are basically visualizing. I will hide the others so that it's a little bit tidier around here. And now I want to play with the pixel classifier. So I will define the background, and this class 1, this will be my mitochondria. Okay. I can switch to black and white. It may be easier for the detection. Now what I want to do, I want to define background. So if you remember the first time you worked with paintbrush, maybe many years ago, this is a little bit drawing we will do right now. So as you can see, not just specifying, or I'm not precisely picking only the background. I'm also drawing across my mitochondria. And the reason why is to help the pixel classifier really distinguish our objects. So now when I have some of the background, let's look at the mitochondria. And now it becomes more artistic, more precise. So I will tell him this is the mitochondria within the background. This one as well. And let's simply draw around few of them. If the image is variable, if there are different intensities, different cells, it's of course beneficial to mark the cells or the situations which are non-standard. So for example, I see there is a very weak signal over here in the corner, but obviously these are still the mitochondria, so I will mark them as well. Maybe I will also specify the background here. Let's do it like this. This is background, and inside are my mitochondria. And after a while of painting, what I can do, I will zoom out so that I could see it. And if I leave the software for a few seconds, it will start applying the learning model. So I will see first preview of the quick results. And I would actually say this already looks very good on my first attempt. So what I will do is now click on full training, and now Mica is executing, or the software is executing the full training. I will save it, my mitochondria save, and now I can actually proceed with the real analysis. Let's go to results, and now I will just apply the model I just learned to this image. So load it from the image. No, I wanna have started like this. So we have this one, yes. We will work on the mitochondria channel, and we will load our model, which is called my mitochondria. And this is pretty much it. I can start batch. And now the software is applying what I just taught it in the previous step. And now we already get some results. So this is area and the total count of mitochondria with this area. So obviously there is more of those which are smaller. I can, of course, add other parameters, length, width, and have other graphs, for example. What I can do further, I can, for example, make a selection of mitochondria of specific length. So, and now once I select it, I can view them very nicely already in the image. I really like this, and I can actually take it into another step, and I can create another graph, set gate. So I will now create another scatter plot just with these, and now I can play further with the parameters. So for example, I don't know, intensity average over area. So these were some example of the scatter plots, histogram. This was total count. There are, of course, other parameters like what is interesting is roundness, of course, because some of them are small and very round. Some of them are elongated. So I'm getting some kind of projection already. If I can get rid of those, I'm not so much interested in maybe. And then there are options for box plot, for example. So I can do area, I can make the scales normalized, logarithmic, and then I'm getting my box plot. So these are just like kind of random examples of the statistics and possibilities we do have. I can also save this layout, and every time I want to do this kind of analysis, I can already load this layout. So this will be my mitochondria. Okay. And there are other functions. I can, of course, save those protocols. I can share the learning. I can export my the models I taught. I can export them, and they can be applied to another Mica software or another Mica station, for example. I can add images to train it even more, to make it more precise. So really, within few, I did not count the clicks, but maybe few dozen minutes from me walking in the room with the sample, just knowing what quality I want to image and what is there. To image analysis, I can move very, very, very quickly. So with this, I would hand the word over to you. We are ready for your questions. We will be very happy to answer them. Let us know, what do you think about Mica? Do you think this make your life easier, or are you interested in seeing it in real life? Of course, we are doing workshops, demonstrations, so all of these are possibilities, and there will be plenty of chances to see Mica live throughout the year. And maybe one question for you, like, what would you do if your microscopy, if your imaging was simpler? What can you do with the time you saved? You don't have to spend so much time training. You don't have to spend time optimizing the experiments. What can you do with the time now?
Hi everyone, that's the presentation over. We now move on to the Q&A session. Both myself, Selçuk, and Svenja are here live, so please ask away your questions. There are plenty of questions already written to the chat box. We'll try to answer as much as we can. So, Svenja, I want to start with the questions. First question, is the incubation mandatory?
Honestly, no. So for sure, it's nice to have an incubation chamber if you do live cell imaging, but if you're just looking at slides, so there are different models of the Mica. So first of all, we have a white film system, and I don't know if you saw it in the Sasha's presentation. So we have in the backpack a kind of backpack that is put behind the Mica. So this is the incubation chamber, and this can be also installed later. So if you are just interested in the Mica itself, and you would like to add your environmental control later, this is mandatory. And also the confocal part is mandatory. So if you say, oh, I'm just interested in a wide field system, but in 2 years you would be interested in a confocal, also this is a part that can be adapted later on. So the environmental control chamber can be installed by a service technician in the field, but for sure, for the confocal part, the system has to go to the factory to be installed, everything like the lasers and so on and so forth. Yes. So a lot of possibilities, so not a lot of possibilities, but at least 4. So wide field with or without environmental control, and wide focal, so wide field and confocal together with environmental control, or even without environmental control.
Thank you so much. Another question asks that, how do you acquire 4 images in parallel?
Okay, so Mica incorporates the FluoSync, and this is our patented, the Leica patented technology. And FluoSync capitalizes the power of the phasor-based and the linear unmixing. And by using the phasor analyzers, each pure spectrum, so whether you have a DAPI, blue, green, red, whatever spectrum, will fall into a defined space of the phasor space. So with the 4 light sources, we excite the sample. So in wide field, we have 4 LEDs corresponding to the most common stainings. For confocal, we have 4 lasers exciting the sample. And on the detector side, we have 4 detectors. So altogether 4 detectors. For each spectrum, we have 1 detector. So 1 camera for each spectrum, or one hybrid detector for each spectrum. So we have altogether 4 wavelengths, 4 lasers, 4 detectors, so 4 cameras, and 4 hybrid detectors. So and the hybrid detectors are the same as in our Stellaris confocal system. So you have finally a real good system, a real confocal.
Thank you for the answer. By the way, there are nice comments between questions. Somebody says that very nice machine. Thank you so much. Next question is, can you combine it with microfluidic chamber for chemical perturbation screens of organoids and/or spheroids?
Yes. So first of all, I think, as my colleague Sasha also showed, you can add it manually via this small chamber that you can slide to the side. But we also have an access, so a kind of blind cap, where you have access to the inner environmental control chamber, and then you can automatically use this for perturbation or whatever, and to treat your samples or your organoids to grow them to specific organoids and spheroids. Yes. So, and finally, just to tell you, so my colleagues did this sample with these 1,000 cells, and they grew the 1,000 stem cells to these embryonic bodies, finally to the organoids, and they had that experiment for 3 weeks. They had that, so they let it run for altogether 3 weeks in the Mica environmental control chambers. So they just use it as an incubator, but together with the usual and continuous checking of the samples.
Thank you so much. Another question, how many slides can I put on one holder?
So at the moment we have a holder for just 1 slide, but beginning of next year we expect to have a holder as well for 4 slides. So finally we have the possibility for 4 slides, but furthermore we have the possibility for the multi-well chambers, so all common multi-well chambers you can put on the microscope, and we have different inserts for different sizes of the Petri dishes.
Okay.
So I think more or less everything that the people have is usable.
Okay, thank you so much. And next question, for live cell time-lapse experiments, which autofocus tools are available for monolayer cells?
So if you set up an experiment, you are asked what kind of autofocus or what kind of cells you have. For example, if you have adherent cells. So adherent cells, I hope you see it. So if you have your cells here, and you always measure the distance between your cover slip and the objective. And if you have a sample which looks like that, the objective will follow. And for this, we have a hardware autofocus. So for monolayer cells, you will be always in focus because the cells that are lying on your cover slip are always next to the cover slip, so that you can, always in focus with adherent cells. But so maybe if we also just were asked about other cells like spheroids or so. So these are more growing up and down, like hills and valleys. So for this, we would have the possibility to establish a focus map so that you follow your hills and valleys on your slide, or if you have a folded slice, that might also be possible, that you can adjust this focus map to be always in focus. And so the last but not least, we also have sometimes confluent cells, which are going up and down. And for these cells, we also have a software autofocus. So maybe you are doing a time lapse, and sometimes the cells are more up and sometimes a bit more down, so that you can also follow with the software autofocus by checking the best for the best contrast and to find the best focal position, depending on where your cells actually are. So we all have everything together. So we have a hardware autofocus to keep the slide in the same position as at the beginning of your experiment. You can establish a focus map, and we have a software autofocus to follow dynamic floating of the cells.
Thank you. Next question. I do image brain organoids sections imaging. How many channels it can do?
So if you have organoid sections, so how many channels? I think that is the same question as before. So we have 4 excitations and 4 detectors. So you can work with maximum of 4 channels and 4 stainings. Do you understand that this is the same? I think that was the question. How many stainings can I use?
Maybe it is specific for the brain organoids, but yes, you explained it. Thank you. And next question is, do we have to use specific consumables to do confocal related applications?
Yeah, for sure. With the confocal, you would like to get the best out of your sample. So you are looking for the best resolution, and therefore it is always recommendable to have thin cover slip thickness cell bottom. So we recommend, there are some recommendations from our side, but honestly, to my experience from my workshops, so if you have thin cover slip thickness bottom dishes, so the ones from EBD, for example, these ones are very good. They are very nicely and precise produced, that you are always in focus, and that you can work with the objectives which are corrected for cover slip thickness of 170 micrometers thickness. So this is recommendable if you would like to really get the confocal quality. And on the other end, if you work with the thick plastic dishes that some people have for the cell cultures, we can work up to 10x, up to the 10x objective, to 10-fold magnification.
Thank you, Svenja. The next question it's about software. Can the exported raw image files be opened in another analyzer software? Or what are the conversion capacity of the Leica software to make it compatible?
So to speak honestly and very simple, so everything, so most people in the scientific world work with Fiji or ImageJ, and ImageJ is able to open these files as well. And for example, so if you would like to analyze the samples and analyze your data, we also, I don't know if everyone knows, so we also have additionally to the incorporated analysis software in the Mica, we have a more complex and more sophisticated analysis tool and software which is called Avia. And if you are keen on using machine learning or even deep learning algorithms, so I would like to suggest you the Avia software, which is also able to use these Leica files, but also all other files which can be used with Fiji for all analysis. And the Avia software would be my recommendation for higher sophisticated analysis. So maybe you can tell more than exactly to your customer, Selçuk.
Okay, and we have a couple of questions more. Another question asks that, is there any risk of radiation toxicity for living cells?
Is there a what?
Is there any risk of radiation?
I just see the question as well as, is there any risk of radiation toxicity? Well, radiation toxicity, so no cell likes to be excited in the UV, like our skin also doesn't like to be excited with UV light. So finally, it is not toxic, but the cells will bleach with the UV light as always. The cells will die if you excite them quite much and quite hard with the UV light. But as a lot of people use the DAPI staining as well, so a lot of people also use it, and well, you will see if the cells die because you excite them too much with the DAPI. So in principle, it is always recommended to use dyes which go further to the red channel, to the red light. So near infrared, for example, so 647 is a typical staining, Alexa 647, and this is recommendable if you really would like to go for lifestyle imaging. So therefore, I would skip the DAPI channel and the UV excitation for lifestyle imaging, and rather start with the green light or with the blue light to excite the green colors like GFP or something like this. To not to harm your cell, but to keep them live as much as possible. And furthermore, so you can change, you can choose in the Mica if you would like to go for best quality, so to excite the cells as much as possible and to get the best out of your sample. Or on the other hand, you would like to go for sample protection, and with this slider, you select well how long the cells will live. So if you are working with living cells, with the slider, you can adjust. And if you go for sample protection, the light will be diminished to a minimum to get a nice image. So this is also one point. So you don't think about how much intensity can I use. You just use the slider and go for sample protection, and then you should be on the safe side and not to bleach your cells and to force the cells to die.
Thank you, Svenja, so much. We have very limited time, so it is last question. Does it wipe or clean the substrate surface when switching away from water immersion objective?
No, you don't have to wipe it away. So for the water immersion, we have an automated immersion. So there is a pump and there is a sensor, and the sensor recognizes if there is enough water. And if there is enough water, it stops, and then you can go live and have a look at your sample. And if you go away from the 63x objective, maybe to the 10x for the next sample, this water drop is sucked, and you can just take away your sample. You're just going to a loading position to easy access your sample, but you don't wipe anything away. So it is really comfortable and really nice, and because it's just water. So you take it out your sample, you just wipe away the water drop which is underneath your sample, and that's all. So you don't have to wipe away this water drop inside of the microscope, because this one is sucked.
Thank you. I guess we are out of time now. So thank you for the presentation and the answers, Svenja, and there are
Thank
still
you.
many questions in the chat box. I guess we can answer them later.
Yes, I think we will answer them by email or by chat, I think. And
okay,
everyone should get its answer. So thank you very much all for your attendance and for the lots of questions. I just see here we have still a lot, and I think we will answer them as soon as possible and let you know. Thank you.
Thank you all.